Welcome, students, to today's lecture on Wilson's disease, a rare genetic disorder that affects copper metabolism and leads to a wide range of clinical manifestations. In this comprehensive lecture, we will delve into the complexities of Wilson's disease, discussing its molecular basis, hepatic and neurological manifestations, diagnostic challenges, treatment options, and long-term management strategies. By the end of this lecture, you will have a deep understanding of this fascinating disorder and its implications for patient care. So, let's begin our exploration of Wilson's disease. Wilson's disease is caused by ATP7B gene mutations that disrupt copper transport, leading to its accumulation in the liver and brain. These mutations can be missense or nonsense, causing dysfunction of the ATP7B protein and the clinical manifestations of the disease. Copper is important for many bodily functions, but too much or too little can cause problems. Wilson's disease is a condition where copper builds up in the liver and affects the body's copper balance. Proteins in the body help regulate copper levels, and when this balance is disturbed, it leads to the symptoms of Wilson's disease. In Wilson's disease, excess copper accumulates in the liver, causing oxidative stress and hepatocellular injury. This can progress to steatosis, fibrosis, and cirrhosis. Hepatic stellate cell activation and pro-inflammatory cytokine release contribute to fibrosis and cirrhosis. Early diagnosis and management are crucial in preventing liver failure. Neurological symptoms are a hallmark of Wilson's disease and often precede hepatic manifestations. The brain is highly susceptible to copper toxicity, particularly in the basal ganglia and other regions rich in dopamine receptors. The pathophysiology of neurological symptoms in Wilson's disease involves the accumulation of copper in astrocytes, disrupting neuronal function and neurotransmitter balance. Excess copper leads to the generation of reactive oxygen species, which cause oxidative damage and neuronal death. Clinical presentation of neurological involvement can vary widely, ranging from movement disorders, such as tremors and dystonia, to psychiatric manifestations, including depression and psychosis. Identifying these neurological symptoms and their progression is crucial for early intervention and improved patient outcomes. Diagnosing Wilson's disease is challenging due to the variety of symptoms and similarity to other diseases. Clinical features, laboratory findings, and genetic testing are used to confirm diagnosis. Early detection is crucial for effective treatment and preventing complications. In Wilson's disease, copper chelation therapy is essential for reducing copper buildup. The main chelating agent, D-penicillamine, forms stable complexes with copper and increases its excretion. Trientine and zinc acetate may also be used depending on the patient's needs. Monitoring and compliance are vital for successful treatment. In advanced Wilson's disease, liver transplantation is necessary for patients with cirrhosis and liver failure. Indications for transplantation include severe liver dysfunction and poor quality of life. Survival rates for liver transplantation have greatly improved, but ongoing medical management is crucial. Neuropsychiatric disorders in Wilson's disease present with various symptoms due to copper accumulation in the brain. Mechanisms involve oxidative stress, mitochondrial dysfunction, and dopamine imbalances. Management includes medications, psychosocial support, and cognitive rehabilitation to enhance patient outcomes and quality of life. Kaiser-Fleischer rings are a hallmark ocular manifestation of Wilson's disease, resulting from copper accumulation in Descemet's membrane of the cornea. These pigmentary rings appear as golden brown or greenish yellow encircling the cornea, visible on slit lamp examination. The evaluation of Kaiser-Fleischer rings involves a detailed ophthalmological examination and, if necessary, corneal biopsy. Copper quantification in the rings helps in assessing disease severity and progression. Treating Kaiser-Fleischer rings involves addressing the underlying copper imbalance through copper chelation therapy. Although the resolution of Kaiser-Fleischer rings may not always occur with treatment, stabilization of ocular symptoms and prevention of further corneal deposition are important goals. Hepatocellular carcinoma, HCC, a primary liver malignancy, is a rare but serious complication of Wilson's disease. Copper overload and genetic factors play a synergistic role in the development of HCC in affected individuals. Excessive copper levels promote oxidative stress and DNA damage, predisposing hepatocytes to malignant transformation. Additionally, specific ATP7B mutations have been associated with an increased risk of HCC development, suggesting a genetic predisposition to malignant progression. Early detection of HCC in Wilson's disease is essential for timely intervention and improved outcomes. Regular surveillance with imaging studies, alpha-fetoprotein levels, and periodic liver biopsies are recommended in individuals with advanced liver disease or specific high-risk genetic mutations. Advances in our understanding of the molecular mechanisms underlying Wilson's disease have opened avenues for the development of novel therapeutic strategies. Targeting specific molecular pathways involved in copper transport, oxidative stress, and neuroinflammation may offer promising treatment options. Experimental approaches, such as gene therapy, RNA interference, and modulation of copper-related proteins, are being explored to restore copper homeostasis and prevent disease progression. These emerging therapies hold the potential to revolutionize the management of Wilson's disease and improve patient outcomes. Wilson's disease in children and teenagers brings unique challenges in diagnosing, treating, and managing the condition. Diagnosis requires considering a broad range of symptoms, and genetic testing is crucial. Treatment involves adjusting medication dosages, ensuring adherence to therapy, and closely monitoring disease progression. Involving multiple specialists and providing ongoing support is essential for optimal outcomes. In conclusion, Wilson's disease is a complex condition influenced by genetics, molecules, and clinical factors. Understanding gene mutations, copper metabolism, and clinical signs helps with diagnosing and treating patients effectively. Ongoing research may reveal new treatment options. Thank you for watching.